Welcome, everybody, here in the auditorium and our international Zoom audience to the 2022 Ogawa-Yamanaka Stem Cell Prize Award ceremony. My name is Deepak Srivastava, and I'm the president of the Gladstone Institutes here in San Francisco. And it's my pleasure to be able to do this event again for the first time since 2019. Because we held off awarding this prize for the last 3 years because this day in person has been so meaningful both for us and for the recipient that we did not want to do this virtually and waited to do this. And I think that's been absolutely the right choice. This is a wonderful day for Gladstone, for the recipient, and for the international stem cell community. This is one of the most significant recognitions for stem cell scientists across the world. And I want to start off by thanking the selection committee who have participated in this award for many years. And those include myself, as well as George Daly, the Dean of the Medical School at Harvard, Hideyuki Okano, Dean of the School of Medicine at Keio University, Lawrence Studer at Sloan Kettering, Fiona Watt in London, and Shinya Yamanaka, who is an investigator here at Gladstone and at CIRA in Kyoto. And we've had the privilege of bestowing this award through the generosity of Hiro and Betty Ogawa and their family. And they have been a longtime supporters of our work here at Gladstone and have both passed in the last several years, but their sons, Andrew and Marcus, continue to be involved with us. And Shinya will say a few more words about the family and this award in a moment. We've also been very pleased to have a partnership that we developed with Cell Press for this award since 2019. And I'm grateful to Debbie Sweet from Cell Press for making that possible. Now, this award, as Shinya will tell you a bit more in a moment, came about to recognize scientists who were pushing translational work using cellular reprogramming for the betterment of humankind. And Hiro and Betty and Shinya had become very close, and they really wanted to honor the field and honor the remarkable work that Shinya had done, for which he received the Nobel Prize in 2012. And so at this time, I'd like to introduce Shinya to say a few words. And by way of background, as most of you know, Shinya actually trained as an orthopedic surgeon initially, and then came to the Gladstone Institutes in the mid-1990s to learn how to do science. So he was a postdoc here in those early days and went back to Japan, did his beautiful work, and we were fortunate to have recruited him to come back 15 years ago and start a laboratory here at Gladstone in 2007, and he continues as a senior investigator now and is now director emeritus at CIRA at Kyoto. So Shinya, please, it'd be great to get a few words from you.
Yes. So this beautiful couple, Hiro and Betty Ogawa, they were very good friends and also mentors of myself and my wife, Chika. Hiro was a very successful businessman with great, huge support from Betty. He actually was the first Japanese businessman who made his own company public in the New York stock exchange market. So it was a remarkable achievement. Then he spent most of his energy to give back to society. So he supported many, many organizations, such as Red Cross, US-Japan Council, Asian Art Museum here in San Francisco, and also the San Francisco Opera. Hiro and Betty were also a huge supporter of science, including Gladstone Institutes. That's how I met with these wonderful couples. So one day in, I believe it was in 1994, they spent a couple weeks in Japan. They enjoyed beautiful cherry blossoms. And before they came back to San Francisco, they came to Kyoto, to my office. And all of a sudden, they started saying that they wanted to have an award with the name of both of us, Ogawa and Yamanaka. I told them, well, you should just have your name, Ogawa. But they insisted, I insisted. So then I said, okay, I'll be back in San Francisco in 3 weeks. So let's discuss this again. But all of a sudden, Betty became sick, very sick. And it was probably like 2 hours. I landed at SF4 in May. She passed away. So this became one of her last wishes. So that's why we now have this award, Ogawa-Yamanaka Award. As Deepak mentioned, this award is for scientists who made transformative innovation in stem cell or research, especially translational aspects using reprogramming. And they made it very clear. They want to celebrate not only scientists, but also their spouses who support the scientist. So that's the most important aspect of this prize. And this year, too, we have a perfect person for this award. And Betty and Hiro, they have 2 beautiful sons, Andy and Marcus, and 3 beautiful grandchildren, Tina, Asina, and Leo. And today, we are very lucky to have Andy. So I would like to ask Andy to come up and to give us a few words. So Andy, please.
Thank you, Shinya-san, for explaining the history and the warm relationship that you had with my parents. I'm sure my parents would be very, very happy right now. I'm sure they would wish they could be here as well. But on behalf of the Ogawa family, we're very, very proud to support this prize and support the scientists and the researchers who continue to work a stem cell to improve the world. So please continue your efforts, and we look forward to continuing to support Gladstone stem cell scientists into the future. Thank you.
Thank you, Andy, and thank you, Shinya, for your comments. So at this time, it's my pleasure to introduce our recipient this year, Juan Carlos Izpisua Belmonte. And the selection this year, I think, highlights a number of things that we value in science, but also in other aspects. And one of the things many of you know, we make a lot of effort towards here at Gladstone is helping bring those who have been underserved populations and underprivileged populations in science to help them develop a career in science. And I mention that because Juan Carlos has a very interesting background. He grew up in a small rural town in Spain, where he went to school for a year or 2 as a young boy, and then helped the family make ends meet, working to support the family. And it wasn't until he was 16 that he was able to go back to school and went to high school. That's really remarkable. And here we are today. It's very, very meaningful. It was there in high school that he met Conchita Rodriguez Esteban, who's here today, who later became his wife and partner, as I'll mention later. I want to also mention that the high school that he went to when he was 16, after not having any education, is now named after him. He went on to university to get his bachelor's degree at University of Valencia, and then a PhD degree at University of Bologna, and subsequently did a postdoctoral fellowship with Dennis Dubois in Heidelberg, where he did really seminal work in the late 80s, early 90s on organogenesis, particularly on limb formation. And it was one of those periods of explosion in science of really understanding the Hox genes and how cells are adopting their fates and forming structures. And it's that theme that he continued throughout the rest of his career, and particularly for the next 30 years at the Salk Institute, where he was from 1993 to until this year. And it was during that period that he has made seminal contributions to understanding how cell fate decisions are made, how organs are built, how one can make chimeras to help create human organs in other animals, do stem cell reprogramming, and most recently, the area of partial in vivo reprogramming that I'm sure he'll talk more about, and which is a significant basis for where he is now, which is with the Altos Laboratories that many of you have heard has several branches around the world, including the one in San Diego that Juan Carlos now leads. Over the years, he's published over 500 scientific papers. He's been named as Time's 50 most influential people in healthcare in 2018 and received many other awards. And he's done this in a close partnership with his wife, Conchita, as I mentioned earlier. And Shinya indicated that Hiro and Betty thought that the partnership was very important at many levels. And in this case, Conchita, who herself was a renowned art person in the field of art restoration, when they moved to Salk in San Diego, well, being in a country that's only 250 years old, there's not so much restoration to be done. And she trained in science, got a PhD, and then has been a critical part of the laboratory for many years, as I understand, really supporting the science that's been, beautiful science that's been done over the last 3 decades. So congratulations and thank you for being here. And as a surprise, just about 10 minutes ago, a surprise for Juan Carlos, not for me, because I knew. Luna, his daughter, and her husband, Bennett, just flew in and are here to celebrate with them. And their son, Elias, hopefully is joining online as well. So Juan Carlos, we're delighted to have you as the 2022 recipient of this prestigious award, and to really look forward to your comments and summary of your work. And at this time, I would love to have you come up along with Shinya and Andy to give you the prize. And before you do that, let me just say a word about the prize. So the prize is a $150,000 gift from the Ogawa family, which the recipient receives. But I think equally important, we worked very hard to make the actual trophy for this that you see here, which is a beautiful crystal, quite heavy piece, as you'll see, that a local artist made, and is at the top of it is we've made a Waddington diagram that maybe Juan Carlos will talk a bit more about, but it represents the epigenetic landscape that we all refer to when we think about cell fate and cellular reprogramming. So at this time, if you all could come join me on stage for this award. Great, congratulations.
Thank you very much. Thank you. Thank you. Thank you. Thank you. Well, good morning, everyone. It's really an honor for me to share this day with all of you. I would like to express my most sincere
and heartfelt gratitude to the Ogawa family, Hiro, Betty, Andrew, Marcus, and the entire family. To Shinya, thank you very much. To Deepak, the Gladstone Institute, to the selection committee, as well as CellPress. This is recognition and an award, and your support means a lot to me. I would say that, just simply say, this is one of the most important scenes of my scientific career. And I am really thankful to everyone who somehow made this possible. Thank you very much. So I'm in thinking, what do I tell you? I thought that maybe the best will be to give you a brief overview of some of the experiments that we have done in the lab in the last few years, but all of them are led and inspired by the work that Shinya did many years ago, around 15, 16 years ago. This paper really changed our way of thinking in the field of developmental biology and many other fields. And personally, for me, was a key moment in my scientific career. So I thought I will just mention a few of the experiments that are based on this work that in my mind has revolutionized biology and probably will and is already doing that in the field of medicine. So, and I start with this. This is the way we all start our life when the first two cells, the oocyte and the sperm, get together. And then an embryo appears after a few weeks that you can see there in the slide. And 9 months after, this beautiful person just come out, which, by the way, is my daughter here. And then in the next, 30, 40 years of our life, we have this explosion of life energy that, unfortunately, with time, not that unfortunately always, but with time, that energy and that stamina start to disappear. We get to an old age and then we all disappear. And that's all our life. And it seems that it's always one way. There's no way backwards somehow. And if we try to represent this at the molecular level, you can see here cells in blue that have a trajectory during our life, but every now and then these healthy cells start to deviate from this trajectory and disease appear, many diseases that end our life. And the key question that I would like to discuss with you today is whether we can change these disease trajectories at the cell level. Well, this one-way street, it's something that can be reversed. That's my main interest, trying to see if that's possible and understand better this phenomenon. Deepak mentioned the design of the award this year, this beautiful crystal piece. And based on the landscape that it's known today as the epigenetic landscape of Conrad Waddington, a very influential person at that time for many areas of biology, but in particular in developmental biology. And he said one thing at that time, that was 57, 60, 65 years ago, that mammalian development is just one way. That when a cell has decided what to be and is determined to do that, like when you want to be an engineer, a biologist, you cannot change that anymore. So this cell fate cannot be reversed or transformed, just one way. Where we are now 65, 70 years later, and thanks to the work of so many scientists, we know that that statement is not exactly true, that there are ways by modifying the genome, the epigenome, factors that are outside the cell, that cell fate can be changed. That is not totally determined as Waddington told us at that moment. But perhaps the key experiment that has demonstrated this concept better than anyone else is these two experiments that John Gordon and Shinya did several years ago. The ability of taking a cell of an animal and then put it into a nucleated frog egg and get an entire frog. So that cell that was going to be an intestinal cell now can be redirected to make any cell of our bodies. And then Shinya that was able to demonstrate that this could be done with just 4 transcription factors. And this obviously has opened up the entire field of biology, medicine, because now we can look both ways. It's not just one way how our life has to move forward. And so this has led to almost just that particular experiment to almost an entire new brand of medicine, regenerative medicine, on how this conceptual discovery can lead to the creation of cells, modification of cells that can be used to treat and alleviate disease, the field of cell therapy. Now, where do I come in from here? My background is in developmental biology, trying to understand how do we go from these 2 cells, the oocyte and the sperm, to that beautiful girl that I showed you before. And we start this way where we see one embryo cell start to divide in 2, 4, 8, and so on. And after a few days, you start to recognize the first embryonic stem cells that will give rise to all the cells of our body. And these cells organize in the space and give rise to this structure, which is the embryo where an organism, human being, will come from. Now, that has been my background, my entire life, trying to understand what are the cell and molecular bases of this phenomenon. But it was thanks to the work of Shinya that these initial insights that we were getting from studying embryos in the lab were greatly improved, because this in my own lab, in my own life, led me to the idea of generating by this reprogramming technology, not just cells, but even structures like the one here shown. This is a kidney organoid, a mini kidney, that it was at that time one of the first, that was 2010, the first organoid that appeared, just a small structure that tried to recapitulate what organs are, starting from an adult cell, and thanks to the technology of Shinya, that we could do these things with human cells. Or maybe the creation of organs inside other animals, the creation of human organs, this idea to replace organs that are failing. These experiments with chimeric cells between different species, human, monkeys, pig, and rodents. And even more lately, just the possibility starting from a skin cell, as Shinya did, to generate synthetic embryos without the need of starting with the sperm or the ovary. So this has been possible thanks to this initial discovery of Shinya. And I have been very blessed and very happy to get busy with this experiment that I could not do before and I could not even dream of doing before. Now, in addition to the possibility of generating organ cells that can be used for transplantation, another curiosity of mine is the ability that all animals have to regenerate or try to make better function when the organ or cell function goes down. So what are the internal endogenous regenerative mechanisms that make that possible? We all know that there are animals that do this beautifully. The Mexican axolotl is perhaps the champion of all regenerators. The zebrafish, the salamander, if you cut the limb, I hear something, you cut the limb of, for instance, the Mexican axolotl, you can see that in approximately 3 weeks, 4 weeks, the limb just get the same as before. It's an identical limb, and this is really incredible. Now, we have learned in the textbooks that we mammals are not able to do that we don't have that capacity, but wait a second in the next slide. So how these animals do this? For instance, let's take the heart of a zebrafish. If you cut it in about a month, then if you don't kill the zebrafish in the operation table, the heart will come back. You can even cut until 35% of the ventricular mass. And the question we ask ourselves is where this regenerate comes from. This new piece, does it come from cardiomyocytes that are present in the remaining of the heart, differentiated cells, or there are progenitor cells inside the heart that will differentiate and will make for the regenerate? I'm not going through the details of how we did this, but the answer is that this new piece comes from adult cardiomyocytes that they have now somehow go backwards and change. And you will see they don't change exactly their fate. What they do is that they normally, a cardiomyocyte is a cell that does not proliferate. Here you have the normal cardiomyocyte, especially this sarcomeric structure that you can see there. Well, when the zebrafish start to regenerate, what happens is that the cardiomyocytes go one step backwards in their history, so to speak, but now they can proliferate. Before they couldn't, now they can proliferate. And so they don't lose their identity as cardiomyocyte. Still, they look like a cardiomyocyte. You can see the arrow, the sarcomeric structure there. They don't go back to an iPS cell, to an embryonic stage. They don't use Shinya's technology. They differentiate a little bit, but then they are able to proliferate, and that's what made the new heart. So as I said, when we went to college, we were told that mammals do not regenerate. That's not exactly true. During embryogenesis, almost any structure can be regenerated, and even after a few days after mammals are born. So this is a mouse of 1 day old, and it has 2 kidneys at that time already. And if you cut one of the kidneys and wait for a week, and you don't know which one, you don't label them, you will see that the kidneys are regenerated. And that's a mammal, and you cannot distinguish the one that was cut versus the other that is not cut. And we know that these things happen in other organs, like, for instance, the heart or fingers and many other structures in our body. So regeneration is present in mammals. The only thing is that it diminishes with time. As we grow older, that capacity to endogenously regenerate get lost and lost and lost. Now, let me pause a moment here. So I've told you that my interest is trying to understand how from a single cell you differentiate into the many cell types of an organism. I have mentioned about the importance of the concept that Shinya put in front of us to try to understand this process. Just, well, let's go the other way. And by comparison, maybe we can understand this process better. And then just the idea that there are some animals, even including mammals, very early on in their life, that they have this small dedifferentiation feature. They never go back to iPS, but that's allowed them to maintain functionality when this is lost. Through these many years, we have started to learn what are the molecular processes that go with this loss of ability to regenerate from a young organism to an old organism. And there are many factors. Genomic instability. About 40 to 50% of our genome is composed of these repetitive elements, transposons, that are a big source of genomic instability, and they get highly expressed as we get older. Our mitochondria, they don't work normally. As we get older, accumulation of senescent cells, stem cell exhaustion, etc. And this happens differently in many different cell types, but something that is common to every cell type in this process of aging is the losing of heterochromatin. This is a specific organization of the chromatin that get lost with time. And this equilibrium that is needed for normal functioning of our cells, that get lost as time passes. And this in every cell type of our organism. So the question that from me after seeing Shinya's studies was, okay, let's take an old organism. In this case, it's a mutation in the lamin A gene that causes this accelerated aging syndrome. And what if we were to reprogram these cells? Can we also see changes in the heterochromatin, a key characteristic of getting older? And as you can see here, that's the case. So in these cells from these accelerated aging patients, heterochromatin levels are low because they are old. When you reprogram them to an iPS cell state, then they go all the way back. And then when you differentiate the cell to an adult cell, now again, the heterochromatin levels go down. So that says that this heterochromatin can be restored. Now, the problem of generating iPS, and that's something that Shinya demonstrated as well together with Dr. Yamada, as well as Maria Abad and Manuel Serrano, is that these epigenetic changes that are associated with this reprogramming may drive the development of tumors. And that's not something that we want. And I don't know how, but it was putting all this together, that the zebrafish doesn't make iPS cells, but can regenerate, that all animals get these heterochromatin levels up, that we can change this with reprogramming, that led us in the lab, and here is Pradeep Reddy, that together with Alex Ocampo, that decided, well, why don't we, rather than having the 4 Yamanaka factors all the time on, why don't we just give a short pulse to see if this heterochromatin can be restored without maybe the bad consequences of having tumors? And this is what they did. So here you have a cell that was exposed just to a short pulse of the Shinya's factor. And you can see that heterochromatin levels after 2 days, they go all the way up. And then it changes key components of the cell, like the nuclear envelope of this cell, which is a characteristic of this progeria syndrome, and many other things. That was a key experiment for us. Immediately, Pradeep and Alex went to a human equivalent, an in vivo equivalent of that experiment. They took these mice from Carlos Lopez Otin, who have a mutation in the lamina A gene that that phenocopies this accelerated aging syndrome. These mice, they get old very quickly, and they decide to apply the factors just on the weekend, not during the week, very short pulses of these 4 factors. I don't know why the weekend, that you will have to tell us, Pradeep, not during the days, but it seems they wanted to work on the weekend as well. And here is what they got, that this mouse now, it's a different mouse, and it has increased its ability to regenerate and even become chronologically, biologically younger, as you can see here. Now, if we look in detail, you see that now these mice have less DNA damage, a big problem for the cell to work properly. And even though the mutation is still there, the lamin mutation is still there, somehow these short pulses of 4 factors buffer the damage that is caused by the mutation and allow these cells then to recover and have normal levels of heterochromatin and be in a much better state. Now, if we compare this with an experiment where we just fix the mutation genetically, you can see that the phenotype is more or less identical. And this, to me, is something extraordinary. Here on the left, we are fixing the mutation that gives rise to this accelerated aging phenotype. In here, the mutation is still there. We have just buffered the ability of these cells to respond to the mutation, and more or less the survival time is the same. So one could say, well, that could be something characteristic of this mutation. What happens if you were to do the same thing with normal mice that don't have that mutation? And Pradeep, again, together with Henry, Jasper, and many other people in the lab, did the same experiment in normal mice, and they could see that they respond better to many injuries and insults that we were doing, and no tumors were generated with these short pulses of Shinya's factor. So from then on, we thought, wow, this is great. We need to go to humans and start our first clinical trial to see if we can rejuvenate a human being. And I'm very pleased to say that we got the results, the first result last night, and I'm sharing it with you. And Shinya volunteered to be the first person to do this. And as you can see here, this experiment, it seems to be working somehow. We still have to do a few fixes on Shinya, and we'll see how this works, but maybe this could be an avenue, and I really appreciate Shinya's work, not just for volunteering for this experiment, but really all what his experiment has done for our lab and for myself in particular. Now, I'm telling you only about aging, but this happened to be the first experiment we did, a mouse that has this accelerated aging. But the key question to us was, can this happen in a young situation when you don't have all these? Is this just a technology that can be used to rejuvenate cells, or can it be used to treat diseases, phenotypes that have nothing to do with aging? So a few years ago, we started to generate different strains of mice where we could partially reprogram specifically certain cell types. And I'm going to show you just 3 quick examples. Can these short pulses of partial reprogramming change the disease trajectory that has nothing to do with aging? Well, here is a first example. This mutation is a mutation in the leptin gene that makes these animals eat all the time. And as a consequence, they get really fat. So we apply short pulses of Shinya's factor, and you can see here that the fat goes away. This is not an old mouse, it's a young mouse. And even though the mouse doesn't stop eating, the fat doesn't accumulate. So that stressor has nothing to do with the mutation. This is a different mutation that compared to the progerin one. Now, this could also be repeated in a normal mouse that doesn't have a mutation. You just give the mouse a high-fat diet for a couple of months, apply short pulses of Shinya's factor, and you can see that the mouse loses that fat. And that, again, in a young situation. Let's put another example. This has to do with toxins. We are exposed to toxins every day of our lives. And in particular, this one, acetaminophen, it's very toxic for young mice, young kids, when the doses are very high for the liver. So you can see here in this mouse with a high dose of acetaminophen, after 9 days, all the mice are dead. But if we pre-treat these mice with a Shinya factor, you can see that a big percentage of the mice, 50% of the mice, now they are more able to buffer this toxin and they can survive the treatment. Another example is fibrosis. Fibrosis is something that goes along many diseases. And you can see in this example how adding the 4 factors for a short time, it can prevent the progression of the fibrosis. In this case, is kidney fibrosis. And a final example, an injury in our muscle. Our muscle can regenerate, and they do that much better when we are young. But even at their peak of regeneration, you can see that by using Shinya's factor, these mice have more satellite cells, and being even young, that they can regenerate perfectly. Now they can regenerate even faster than the wild type mouse. So what I want to conclude with this is that this is nothing to do about just an aging phenotype. It's about a healthy phenotype to prevent and maybe treat disease phenotypes, not just in all organisms, but in young organisms. So the question that I posed there is not anymore a question in my mind, at least with mice. And this could be used, if we were to know more, as an approach to treat and prevent diseases. Now, the 2 key messages I wanted to leave you today with is that the one I just said, that partial reprogramming can help to alleviate diseases independently of age, and that heterochromatin, the presence of heterochromatin in these cells when they are not working properly, restoration by short pulses of Yamanaka factors play a key role in this alleviation of the diseases. Now this is a slide I borrowed from my colleague Diego Costa and Rick Klausner. What does it mean? I have put for the first time the word resilient. What is resilience at the molecular level? So let's imagine stress. Stress is this acetaminophen, this high-fat diet, this mutation, or even the biggest stress of all for a cell, just passing of time, aging. So the way that the body responds to that stress, it varies with time. When we are young, this buffering capacity to respond to these all stresses, and when it meets with this stress graphic there, is high up. And then that means that that cell, that organism at that moment has a much higher buffering capacity to cope with any insult. But as time passes, you see that the crosses of the stress level and the capacity to respond is much lower, and that leads to the disease. So ideally, what we would like is to use technologies like this one here, partial reprogramming and many others, to program cells to make them much more resilient. And this is a new concept. It's not treating every specific disease by itself. is trying to increase our resilience, our buffering capacity. So when the insult, when the stress comes, we can cope the same way that we cope when we were young. So in a way, this is a little bit different of how medicine is practiced today. And precision medicine, something that in the last few years is exploding, which is identifying a gene that is, it's somehow whose expression is altered, and that's going to alter the molecular function and the cell function, and in the end leads to disease. Fixing this particular stress, and that can be done by gene therapy, and we know about the revolution that we are experiencing in the last few years, how pharma industry is doing this, specific and traditional drugs that go after that particular problem. But what I'm proposing here today is maybe slightly or quite a lot the opposite. Not to focus on the specificity of that stress and just try to increase the resilience and buffering level of the cell to cope with the many insults and stresses that one can go through our life. But before we can even dream of repeating the experiment we did last night with Shinya, we certainly need to understand how this happened at the cellular level. This black box of partial reprogramming is something that is ahead of us, that we are really excited to try to understand this process better, that maybe one day this could become a reality. And my final slide is again to thank everyone for this award, but very especially the people in my lab that throughout all these years have put forward all the concepts and ideas that I have tried to summarize today. This is a picture that was taken in 2019, so it's a little bit older, but we haven't got together to make the picture since then. But many of the people in these pictures, their work has been key to conceptualize some of the experiments I have indicated today. On the left, there are people who have left the lab, and they have been really wonderful friends and key members of this scientific journey together, like Alex, Juan, incredible person, and everyone else. And here I have the pleasure of having with me today Pradeep, who somehow together with Alex came to this idea of short pulses of Shinya's factor. I would also like to thank May for all these years, more than 2 decades of, when you have a person that can make things happen without noticing that things are happening in the lab. And this is something that it's priceless. Thank you very much, May. And finally, sorry. I would like to thank Conchi, my wife, for all these years of love, support, and inspiration. Thank you very much. And it's really a big surprise having my daughter here. Thank you. Thank you, Luna.
Thank you so much, Juan Carlos. Fabulous summary of your beautiful work. We do have time for some questions from the audience. Maybe I'll start, Juan Carlos. Tell us about how you're thinking about going about this next step and with your move to Altos, and how do you see that advancing this project?
So Deepak is asking me, how is it that I moved and left academy?
I didn't ask that, but go ahead.
And I'm working in a place called Altos. So I never thought I would leave academy. I was very happy all my life trying to goof around with these experiments and see what happened, reading the latest paper from Shinya and trying to see what else we could do. But I used to finish all my publications with more or less the same sentence, which is maybe, perhaps one day, 100 years from now, this picture, this concept that I am showing here in this publication may help someone somewhere. And I got this call from this person, Rick Klausner, former National Cancer Institute director. So is that true? Are you really serious on thinking that maybe one day this paper could become a reality? And that was through the dark days of the pandemic, where all of us, I'm sure, reflected in our past life and how we could be better in the future. And that call was really instrumental to, well, let's try to make this a reality. And how to make this a reality, the idea of increasing buffering capacity in a cell to cope with diseases is a huge challenge. I would say that that's one of the major challenges we could have in medicine today. And just even trembling about formulating that question, can this be done in humans? But it's a challenge that I'm willing to take, and especially with what Shinya put in front of us. There could be many other approaches, many other methods to try to increase this resilience and buffering capacity, trying to understand how this happens at the molecular level. And once we understand this, is perhaps when we can start moving. I think it will take time. Probably we'll have to think of ex vivo applications rather than going to in vivo, and see whether that's possible. It's difficult to foresee the future, but I see a path there based on this experiment we're doing with mice, of this idea that increasing buffering system may help cell health and organismal health. How? I don't know. We have started a new adventure, trying to get the best people we can to work together and make that conceptual discovery that Shinya and many others have now applying already in the clinic. And many of the studies coming out of these institutions are a real proof of that, while expanding that with this new conceptual idea.
Yeah, no, thank you. It makes a lot of sense. And I think we already see so many examples, even in neurodegenerative disease, for example. Even when you get older, some cells can buffer better than others, and there's selective resilience for reasons we don't fully understand. But hopefully we'll get to the bottom of it. Other questions from the audience? Don't be shy, our trainees particularly. Yes.
I was just wondering.
Go ahead, if you wouldn't mind using the microphone so that the online audience can hear you as well.
Thanks for a really good talk. Very exciting work that you've done. So de-differentiation seems to be a very beneficial process. I was wondering if if you're doing this systemically in an organism, do you have any concerns about targeting cells that might be very important in their specialized roles and de-differentiating the wrong cells?
Yes, we're very concerned, and in fact, this happened. There are cells that are more prone to de-differentiate than others. For instance, the liver, which is one of the organs that maintain that regenerative capacity during our life, and it does so by de-differentiating, so to cope with the stresses that goes through. So short pulses of Shinya's factors immediately de-differentiate these cells and make the liver non-functional, which in the end kill the mouse. So that's a major concern. So what we are aiming at, and we feel that we are getting the right approach, is to prevent that de-differentiation. We want to maintain cell identity, in this case, hepatocytes, not de-differentiate back to a progenitor cell or any other cell type, to prevent what you are saying, the big major concern of not having the liver working. There are different cell types. They respond differently to these pulses of Shinya's factor. So nailing down how every cell type, every context in the cell and organ needs where this happens is a major aspiration of ours, trying to prevent that from happening. You have nailed down the key challenge that we have in the field, prevent the cell identity to disappear. In some of the examples I have shown you today, that seems to be the case, but we have to make sure not just in one cell, but in all the cells of that organ, or as you said, somewhere else in the organism, that that's not the case.
Thank you.
Good question.
Great. Well, please join me in thanking Juan Carlos for a wonderful seminar.
Thank you.
Well, I want to once again thank Shinya and Andy and the Ogawa family for this wonderful prize and Cell Press. And I want to note that none of this would have been possible. None of us would be here had it not been for Bob Maley and his wife Linda for taking the brave move to start Gladstone out of nothing in 1979. And we're delighted to have you guys both here today. And finally, I want to note that some of you know that our stem cell center here at Gladstone is named after the Roddenberry family. The Roddenberry Foundation of Star Trek fame have supported our stem cell center for the last 10 years. And it's worth noting that in the original Star Trek movies, there was often scenes where wands would pass over damaged tissue and restore it and regenerate it. And that has been, in fact, their dream. And this is what inspired them to support our center, that someday, through the technology that you heard about here today, we might be able to regenerate damaged tissues. So I know on behalf of the Roddenberrys, they're delighted that you are the recipient this year, Juan Carlos, and hopefully that vision of Gene Roddenberry someday will be reality. Well, this concludes our 2022 Ogawa-Yamanaka Award Ceremony, and thank you all here in person and those online from around the world for joining us, and congratulations again, Juan Carlos.